I'd like to now talk to you about a special kind of gut motility called vomiting. Now, it is an evolutionary fact that when you see someone vomit or hear them starting to retch or even smell vomit, you almost feel like you need to vomit as well. I'm sure you've all had that experience. And by evolutionary fact, I mean back when we all lived in the same cave and we ate the same food, it stands to reason that if one person started to vomit from the food that they ate, probably we all should vomit it up. Maybe it was rancid. Maybe there was some sort of bacterial contamination. I'm going to tell you about the different centers of vomiting control. The major one is in the medulla oblongata, and there are afferent incoming signals from the vestibular apparatus, which is found in your ear. There are also afferent information from the back of your throat. You, I'm sure, have known that once food touches the back of your throat, it's not going to go down well. There are even sensory cells in your GI tract once the food hits there in your stomach or even small intestine that you should probably get that out before you absorb it. And there's also some information sent to the medulla oblongata from the fourth ventricle, which is actually bounded superiorly by the pons and inferiorly by the medulla oblongata. So the fourth ventricle is actually monitoring what might already be in your bloodstream. Say, maybe you drank too much alcohol. Now, vomiting is all about reverse peristalsis. You have to get the food out. In order to do this, you have to have relaxation of the stomach and the pylorus. You can even vomit from your small intestine. You take a deep breath in, that's the forced inspiration, and this is to increase abdominal pressure. You have to relax in the esophagus, the lower esophageal sphincter. This is the band of muscle of your esophagus that just leads to the stomach, so you have to relax that. And then all of a sudden, you have forceful expulsion. You have massive contraction of your abdominal muscles, and this helps push your chyme and intestinal contents up through the esophagus. If your upper esophageal sphincter stays closed, then all the contents went up to that point, and then they return to your stomach. This is called retching. And when you do that, when you bring it up to the back of your throat and then you swallow it back down again, you're very mindful of this, and probably the next time you try this, you're going to relax the upper esophageal sphincter and get the whole thing out. So the next time you're watching your patient vomit, it's not in their best interest to have a nurse that's also vomiting with them. As you're holding back their hair and holding their little tray so that they can vomit into it, I want you to go through all of the steps, all the afferent signaling to the medulla oblongata, and go through all the steps in order for vomiting to occur. Maybe that will help you from vomiting along with your patient. As we continue with the types of motility, we're going to go now into the small intestine. And as I corrected myself earlier, in picture A, it's showing segmentation, which that means there are local areas where there's contraction and then relaxation. Contraction and relaxation. It's a lot like kneading bread dough before you put it actually in the baking pan. So that's called segmentation. But the small intestine is still capable of peristalsis, which is shown in picture B, where of course behind the bolus of food or chyme there's a contractile ring, and ahead of it is receptive relaxation. There's also kind of a pendular motion that occurs, and this is reverse peristalsis followed by peristalsis, so kind of a back and forth kind of movement of the chyme through the small intestine. Now when it comes to the large intestine, there are mass movements, and there are 1 to 3 of these per day. And basically what it means is there's this contractile ring, receptive relaxation, and you move your fecal material closer and closer towards the sigmoid colon and ultimately to the rectum. When the rectum is about 25% full, you get the urge to defecate. Whether or not you do that is up to you. Fact, there are a lot of people who have, how do I say this? They have overridden this urge to defecate. They don't allow themselves to go to the bathroom and defecate. Maybe it's socially unacceptable. Maybe they feel awkward with the company that they are. And this basically means if you do this often enough, you run the risk of becoming so impacted with fecal material and your colon loses its ability to respond to these stimuli that you can no longer defecate. And when you do want to defecate, your feces are so compacted and desiccated, they don't have enough mucus to help them slide out of your body, you become what nurses say, FOS, or full of, I'm sure you can fill in that last letter. And this is a true phenomenon. The most common population to lose this ability to defecate because they're so embarrassed and with their peers so often are toddlers in preschool or daycare or teenagers. They're with their peers for most of the day. They know it's a private moment and they fight the urge back. And when they do that often enough, they get a lazy gut and their fecal material becomes impacted. It is a truly big problem. I have had students in the past who worked with nurses where the nurse's job was to basically reteach the child to become mindful of the signals from their gut and to actually go and try and bear down on the toilet. So all of this is part of the gastric colic reflex or law of the gut. Food in, feces out. Now I feel compelled to tell you that the law of the gut can work in reverse. And the gentlest way to put this is, be careful what you put in someone's anus and rectum, like a thermometer. You need to hold on to it. If you put something in and walk away, there's going to be receptive relaxation ahead of that object and a contractile ring behind it. And it's very easy for things to start moving upwards through the GI tract. And on a social level, there are some populations that have practices where they might insert things into the anus and rectum, and this becomes problematic because of the law of the gut, it gets sucked up further, and then they have a problem retrieving it. I have had many students tell me who have worked in the emergency room stories about people seeking medical attention because a flashlight, light bulb, even a fish sucked up into their rectum and sigmoid colon and they could not retrieve it. So just be careful what you insert into the anus and rectum because this law of the gut can work in reverse. I'm now moving on to secretions of digestive enzymes and buffers, and I've tried to color coordinate things as much as possible. And if you take a moment to orient yourself with the different titles I have on here. I have salivary glands, which of course would be associated with the oral cavity, followed by the stomach, followed by the intestines. Along with the intestines is the pancreas. So in kind of this pink color, I'm highlighting enzymes that are related to carbohydrate digestion. In the salivary glands in your oral cavity, you actually begin carbohydrate digestion with your salivary amylase. If you were to chew on a cracker and just masticate it over and over without swallowing, it would become very sweet to your taste buds. And this is because your amylase is starting to digest the polymers of the carbohydrates into simple sugars. Of course, once the food reaches the intestinal mucosa, we still have more carbohydrate digestion. The intestinal cells will secrete different enzymes like sucrase, maltase, lactase, whose job it is to digest disaccharides into smaller monosaccharides like glucose and fructose. And then the pancreas really releases copious amounts of digestive enzymes, one of which is pancreatic amylase, and this will further digest any carbohydrates into simple sugars. In green, I've shown areas of your body that release enzymes that can digest proteins. Protein digestion begins in the stomach. The chief cells release pepsinogen, which in turn is converted by the hydrochloric acid into pepsin. So protein digestion begins in the stomach. It continues in the intestines. The intestinal mucosa releases peptidases, which that name tells you they break down the peptide bond associated in between amino acids. And the pancreas, again, releases copious amounts of enzymes that can break down proteins. In the pancreas, I have listed here trypsin, chymotrypsin, and carboxypeptidase. I need you to know that these enzymes are actually stored in the pancreas in an inactive form. They are zymogens. The word zymogen means inactive form. So in the pancreas, trypsin is actually made and stored as trypsinogen, and chymotrypsin is chymotrypsinogen, and carboxypeptidase is stored as procarboxypeptidase. This is a similar phenomenon that we have seen already in clotting factors. Many clotting factors were made in a precursor form, and their names either ended in ogen or pro, like prothrombin or fibrinogen. Those are all zymogens, which means they must be activated in order to become an active enzyme. In yellow, I'm highlighting enzymes whose job it is to digest lipids. Now, even though there is a lingual lipase, it has very limited enzymatic activity in the oral cavity. So I will not be testing on that. We're going to pretend like it's not even there because it's very, very insignificant, the contribution it gives towards lipid digestion. The biggies come from the enzymes from the pancreas, as I've said before, through lipase and cholesterol esterase. So these are going to be able to digest your fats into monomers that you can absorb. A special type of intestinal enzyme that is released, shown here in the top right corner, is enterokinase. Enterokinase is really important because when the pancreas releases trypsinogen, chymotrypsinogen, and procarboxypeptidase, it's enterokinase that activates them into trypsin, chymotrypsin, and carboxypeptidase. If it could speak to you, it would say, hey, you don't want these enzymes activated unless I detect food here. So sensory cells will signal the release of enterokinase if they detect food. Once enterokinase is there, then the pancreatic enzymes can be activated, once and only if food is present. The next thing I want to highlight in gray from the stomach is intrinsic factor. Intrinsic factor is released by parietal cells. We've already learned about intrinsic factor. It must be released in order for your ileum to absorb vitamin B12. Again, intrinsic factor comes from the parietal cells. If we don't absorb vitamin B12, we've already learned this can lead to a type of megaloblastic anemia. It's called pernicious anemia. And then another enzyme or set of enzymes that the pancreas can release, they're called nucleases. So you have DNases and RNases. Clearly the food you eat has cells. And inside nucleated cells, we've got DNA. We also have RNA. Why not digest those nucleic acids into individual nucleotides, absorb them, and be able to use them to make new DNA or RNA for your own cells? Let's officially begin our trip through the alimentary canal through the oral cavity or buccal cavity. What are the functions of the oral cavity? First, you definitely assess what you put in your mouth before swallowing. If it doesn't agree with the sensory cells in your mouth, the taste buds, you're not going to swallow. You're probably going to discreetly spit the material out into your napkin, or in the case of young children, they will actually spit it out into their hand and present it back to their parents. There's also, if it's agreeable, there's mechanical processing. You start to chew, you start to roll the bolus into a sphere with your tongue, and also mash it up to the upper surfaces of your soft and hard palate. So you're just kind of trying to mound it up into a swallowable sphere called a bolus. You also are lubricating it with salivary secretions. The salivary secretions, you make about 1 liter a day of saliva. There are many uses for the saliva. It's not just for lingual lipase, which we've already said is negligible. It's not only for salivary amylase, which begins carbohydrate digestion, but there are also antibodies and proteolytic enzymes involved in saliva. Without saliva, within 24 hours, you will start to generate oral sores and cavities. So it's very important. As a side note, if anyone is listening, if you're taking an antihistamine, which is a common ingredient found in allergy medications, this decreases your ability to make saliva. And as such, you are more likely to develop dental caries, also known as cavities. I recently learned this when I started having problems with allergies and started to take some allergy medication, only to find out in about 6 months I had developed 6 cavities when I hadn't had a cavity in over a decade. So watch out for those allergic medications. They are going to decrease your ability to make saliva. Now we're in the stomach. Let's talk about the secretions for digestion and what things can you possibly absorb in the stomach. The stomach, its primary function really is a short-term storage or reservoir for food. And once the stomach secretions start going, we don't call it food anymore, we call it chyme. It's kind of a pasty substance. So what secretions are going on in there? First, to review the anatomy, the part of the esophagus that leads into the stomach, as shown in the top right picture, is the cardiac region, sometimes just called the cardia. The most superior domed shape of the stomach is called the fundus. Then the majority of the stomach is called the body, and then we get to the antrum, or waiting spot, and then we get to the pyloric sphincter, and then the exit through the pyloric sphincter is called the pylorus, and then we get the duodenum. Now we find different gastric glands in different parts of the stomach. We find different glands in the body of the stomach versus the antrum. So if you look at the bottom picture, you can see the most numerous cell type in the body would be the parietal cells, and in green you're seeing chief cells. Then in the antrum you're seeing mucus cells and G cells. And I'm actually going to modify this picture a bit. We find mucus cells even in the body. So along with the parietal cells and chief cells, you should also mark that you would have mucous neck cells. So like I said, there are 2 types of tubular glands that invaginate into the mucosa of the stomach. There's going to be the glands that we find with the body of the stomach and in the antrum. The overall job of the stomach is to liquefy the food. And as I said, the meal that you've eaten, along with the gastric secretions, will suddenly become 1.5 liters to 2 liters, and it will take about 3 hours for that entire volume to exit through the pylorus into the duodenum. So the secretions. The parietal cells release hydrochloric acid and intrinsic factor, and we know intrinsic factor is important for vitamin B12 absorption in the duodenum and ileum, primarily the ileum. Hydrochloric acid is necessary to convert pepsinogen, which is released by the chief cells, into pepsin. Pepsin will chemically digest the protein, the peptide bonds. And then next, we have gastrin. Gastrin is released by the G cells shown in the bottom right picture. G cells release gastrin in a hormonal fashion. Gastrin, released as a hormone, will circulate throughout the whole body, only to end up and target the parietal cells of the stomach to secrete more hydrochloric acid. And then lastly, the mucus cells, shown here in the antrum, but they are everywhere in the stomach. They release bicarbonate-rich mucus, I should say. And this bicarbonate-rich mucus is really important to protect the simple columnar cells of the stomach, so that the very acid and pepsin that are being released and activated don't self-digest the stomach. Alright, so 80% of the stomach is the body and the fundus, and 80% then of the two types of glands that I told you about are the gastric glands. So gastric glands are found in the fundus and body. In the gastric glands, we find three different types of cells. We find the mucous neck cells, and again, I told you mucous cells are found not only in the antrum, but in the body and fundus. And again, as I told you, it helps protect the simple columnar epithelium of the mucosa from the acid and pepsin. If you look in the bottom right picture, you see in purple the parietal cells in this gastric gland. They secrete hydrochloric acid and intrinsic factor, and we need that intrinsic factor in order to absorb vitamin B12 in the ileum of the small intestine. That helps us avoid megaloblastic anemia. Deeper in the gastric gland, we see chief cells. They are the most numerous, that's why they're called chief, and they release pepsinogen. As the pepsinogen moves into the gastric lumen, it's exposed to the hydrochloric acid secreted by the parietal cells, and this converts pepsinogen into pepsin, which can now hydrolyze peptide bonds of the proteins you've just eaten. Now, I don't want you to memorize anything on this slide yet. I just merely want to direct to a clearer picture, I think, on the right side. Both the left and the right picture are showing you the same thing. Start at the bottom of the right picture. Can you see where it says carbon dioxide plus water? Well, the cells of the stomach, the parietal cells specifically, have unique gene expression. They express the gene that codes for an enzyme called carbonic anhydrase. And this is going to be something really important for Unit 4 for proper renal function. So you might want to just hear this just to get it started right now. When you have carbon dioxide and water, your standard byproducts of metabolism, and carbonic anhydrase, the carbonic anhydrase will unite these two into carbonic acid, or H2CO3. This will dissociate, and we know what that means, into hydrogen and bicarbonate. Now the hydrogen is pumped out of the parietal cell into the lumen of the stomach. That's your acid part. Meanwhile, potassium in the lumen of the stomach, probably from the food you ate, is pumped into the parietal cell through a hydrogen potassium ATPase. Then on the side of the cell that faces the blood, we see in green our standard sodium potassium ATPase. It kicks out 3 sodium for every 2 potassium that's brought in. You also see in pink that the bicarbonate that was made through this enzymatic reaction is kicked back out into the blood and chloride is brought in. The chloride will follow along with the hydrogen that's pumped out into the lumen, and that's where we get our hydrochloric acid. The pH of the contents in your stomach are typically around 2, but they can even get below 1, as low as 0.8, and that's phenomenal. When you eat food, this whole process increases, and you get more and more bicarbonate going into your blood. When bicarbonate goes into your blood pH rises. That means you get more alkaline. And when this happens, we call it the alkaline tide. And it partially explains why you start to feel sleepy after a big meal. In the antrum, we see two types of glands, or two cell types in the pyloric gland. This is all in the antrum. We see mucous neck cells again, and we also see G cells. They release gastrin. G cells are enteroendocrine cells. And the gastrin, when it's dumped into the bloodstream, will in turn stimulate parietal cells in the gastric glands to secrete even more acid. And this also will increase pyloric contraction. It will also cause relaxation of the pyloric sphincter. As a result, we get more gastric emptying and more acid production. What exactly can stimulate acid secretion? Well, there are three main phases. Look on the right side of the picture. There's a cephalic phase. Just seeing, smelling, and anticipating food is perceived by your brain. The brain then can tell the stomach to prepare for receipt of a meal. This accounts for 30% of acid response to a meal. This relay from the brain to the stomach is through the vagus nerves. The vagus nerves can also relay back to the brain local afferent information. When you swallow and food touches your stomach, it's the vagus nerve that can relate information back to the brain, which in turn can relate even more secretion. You're seeing the secretory fiber here. Think of that as an efferent outgoing command. Again, all this being relayed through cranial nerve number 10, called the vagus. Additionally, with the gastric phase, in addition to local feedback and vagal reflexes, we see the G cells releasing gastrin, which in turn stimulates histamine stimulation. And histamine, we're going to see, also can lead to even more hydrochloric acid secretion. So that's all part of the gastric phase. Then finally, when some of your chyme touches your small intestine, we have neurons that can detect the presence of food. Also, we detect the presence of food by I cells, S cells, and K cells, which will in turn release their hormones. So all of this will lead to gastric secretion being stimulated by local control, neural, and endocrine. And I've highlighted them according to local for histamine, blue for acetylcholine, which is neural, which comes from cranial nerve 10, the vagus nerve, part of the parasympathetic division, and lastly endocrine from the G cells releasing gastrin. Now, there are factors that can alter how much hydrochloric acid secretion occurs. There's this idea of potentiation, the ability of stimuli to produce a combined response that is greater than the sum of individual responses. Another way of saying that is 1 plus 1 plus 1 does not equal 3. If I consider the local release of hydrochloric acid through histamine, and I can quantify how much hydrochloric acid I get, say a liter, and then I actually collect how much hydrochloric acid I get when I have a neural response through the vagus nerve releasing acetylcholine, as shown in this right picture, maybe I get 2 liters. And then if I actually collect how much hydrochloric acid I get from the G cells releasing gastrin, let's say I get 3 liters. Well, if I get 3 liters for gastrin, 2 liters for the vagus nerve releasing acetylcholine, and 1 liter from histamine, that's a total of 6 liters. But if I stimulate all 3 cells at the same time, or pathways I should say, I won't get 6 liters. I might get 18. So again, 1 plus 1 plus 1 doesn't always equal 3. We get a much bigger combined response. And we've seen this before back in Unit 1 when I showed you hormones that regulate blood glucose levels. And you saw the combined effects of glucagon plus epinephrine versus glucagon plus epinephrine and cortisol versus glucagon plus epinephrine plus cortisol plus growth hormone. You get an escalatory kind of effect. So acetylcholine from the vagus nerve is going to stimulate, of course, muscarinic acetylcholine receptors. And we learned about the IP3 DAG and calcium second messenger system. We also already know that atropine would block these receptors. Histamine is released locally from ECL cells, which stands for enterochromaffin-like cells. They use a different second messenger system. They use cyclic AMP. Drugs like Tagamet, Zantac, they block this receptor. And then gastrin is released by G cells. And the G cells use a different receptor when gastrin binds. But still, it uses a second messenger system, the IP3 DAG calcium second messenger system. So if you look at how two systems use the same second messenger systems, and you already know that second messenger systems are kind of an avalanche cascade effect, you can appreciate why more than one system activated leads to even more acid production. Peptic ulcer disease. All of this acid secretion can be problematic. Again, we're seeing potentiation. We need a good way to protect the gastric and intestinal mucosa from all of these stressors. Hydrochloric acid and pepsin are quite corrosive. Peptic ulcers occur when damaging effects of acid and pepsin overcome our ability of our mucosa to protect itself. If you find gastric ulcers, the main problem is a decreased ability of the mucosa to protect itself. Whereas duodenal ulcers, the main problem is exposure to the increased amounts of acid and pepsin. And in the bottom picture, you're seeing, in blue, the gastroduodenal mucosa, the simple columnar cells. On top of it, in brown, you're seeing the protective bicarbonate-rich mucus from the mucous neck cells. This bicarbonate-rich mucus should deter any of the corrosive effects of the hydrochloric acid and pepsin. So your protective factors are the bicarbonate-rich mucus, good blood flow to the mucosal cells, and we also need the mucous neck cells to be able to express their prostaglandin. Remember, prostaglandin E2 helps them make this bicarbonate-rich mucus. And we also need good growth factors for the gastric and duodenal simple columnar cells to replicate and divide. We need good mitosis. We need to rejuvenate that layer. Anything that damages this, such as hydrochloric acid and pepsin being produced too much, or bacterial infections from Helicobacter pylori, NSAIDs, we've already learned in Unit 1 how NSAIDs are COX inhibitors, and if we do that, we cannot shunt arachidonic acid into the different types of prostaglandins. Stress can also interfere with the bicarbonate-rich mucus synthesis. Smoking can kill the mucous neck cells, and alcohol. So what is this gastric mucosal barrier? It's the bicarbonate-rich or alkaline mucus that resists the acid and pepsin, tight junctions between the simple columnar cells so that the gastric juice can't seep in between the cells and into the lamina propria, which is protein and sugars, and be digested. We need good epithelial cell replacement, a 3 to 6 day lifespan. Good mitotic turnover is what that means. And then physiological. In one of the earlier slides with the postulated acid secretion, Any hydrogen ion that happens to diffuse back into the cell, we need to be able to pump them right back out through that hydrogen-potassium ATPase. If our gastric mucosal barrier is damaged, the biggest problem is the hydrogen back leaks, diffuses back into the mucosa in exchange for sodium. And this is actually the precursor to gastric ulcers. When hydrogen back leaks into the cell's pH declines. This leads to cell death. Cells that are dying through necrosis, they just dump out their contents. And when they dump out their contents, it leads to damaged mast cells, and this means to also damage to the enterochromaffin-like cells, and they start to leak histamine, the distress signal. And it's a vicious cycle. Histamine leads to more of an inflammatory response, which can lead to more vascular damage in the submucosa. Lack of blood flow leads to local ischemia, leads to more leakage of cells and their contents, leads to more acid, leads to more leakage of histamine, and more cell death, and repeat and repeat. Next thing you know, you've got a bleeding ulcer.